So let's look at Mendelian genetics or transmission genetics. The focus here is on pure breeding parents in the parental generation or P generation. The way we'll do this is by looking at crosses between individuals that produce families. So the first cross resulting from these pure breeding lines will be known as the F1 generation, or first family or familial generation, where the F actually stands for filial or family. So hybrids for a particular gene can be produced by crossing two pure breeding parents for a dichotomous trait. This is where, for example, if you have a tall versus a dwarf phenotype for one particular gene, for the same gene, and the dichotomy is the tall versus the dwarf, so that we have tall, and we're going to use the nomenclature capital T, in a diploid where there's a copy of the allele from one parent and copy from the other, versus dwarf, copy of the little t from one parent, copy of the little t from the other parent. This is the way we would get pure breeding lines. Note, in true Mendelian genetics, the F1 provides important information regarding the dominance versus recessive relationship of the alleles of the gene under investigation. So, when we're looking at this, we have to consider if a tall plant that's pure breeding, think about this, is crossed to a dwarf plant that's pure breeding, then the progeny, or the F1, will reveal whether the tall or the dwarf allele is the dominant allele. Because it's not always tall being dominant to dwarf. You could have dwarf dominant to tall. So we would know that the tall parent is pure breeding because we would have crossed tall, crossed to tall, and produce tall progeny. Then we will also look at that the dwarf would have been pure breeding because by crossing the dwarf cross to dwarf, we only going to get dwarf progeny. So when we cross a tall by dwarf, where we get the big T gamete and a little t gamete, and we end up with this hybrid, if the progeny is tall, then the capital T is the dominant to little t allele. If the progeny is dwarf, then the capital T allele is recessive to the little t. Now note, this is using standardized nomenclature that uses capital letters to denote a dominant trait and lowercase letters to denote recessives. But this is not true in all species. Gene nomenclature is species specific. For our purposes, we will be using standard nomenclature. Now consider the following cross. If you have a big T crossed to a T prime, now we have to consider the phenotype of each parent and the alleles involved in this cross to determine which is dominant versus recessive. When we begin to look at the deviations from standard Mendelian genetics, it will become even more important to pay attention to the F1 phenotype, because that's where a T prime, we have to look at that F1 to see whether the T is dominant, T prime is dominant, or whether there's incomplete dominance or codominance. So we have to pay attention to that F1 phenotype. Thank you.